Thank you very much for the invitation to speak at this HBV immunotherapy session today. I'd like to present some of our recent work developing a novel T-cell vaccine for HBV immunotherapy in partnership with Vaccitech, which is a spin-out company from the University of Oxford. I'd like to start with some of the lessons that we've learned from our work in HCV vaccine development. Here is my disclosure slide. So my laboratory at Oxford has worked on developing T-cell vaccines against hepatitis B and hepatitis C virus for many years now. And the rationale for developing T-cell vaccines is that after acute infection, we know that a proportion of people develop persistent infection whilst others resolve infection. And a combination of HLA association studies, CD4 and CD8 T-cell blocking experiments, and the temporal association of viral specific T-cells with infection resolution has highlighted the important role that T-cell immune responses play in resolving infection. So in order to maximally generate viral specific T-cell responses, we've elected to use a viral vectored approach using both chimpanzee adenoviral vectors. Everyone has now heard of these following the success of the Oxford AstraZeneca COVID vaccines, but also modified vaccinia Ankara vaccines, MVA, which can be used together in heterologous prime boost strategies. For hepatitis C vaccines, we've encoded the non-structural proteins from a single genotype 1b strain. And to begin with, we applied these vectors using two different adenoviral vectors in heterologous prime boost strategies in healthy volunteers, as shown here in the purple line. And the data here is an interferon gamma ELISpot assay that measures the magnitude of the HCV specific T-cell response. And you can see that after the first vaccination, there was a robust response to the adeno vaccine, but after the heterologous adeno, there's an attenuated response. The most likely reason for this is that there's antibodies to viral vectors that inhibit the second vaccine. And for this reason, we replaced that heterologous adeno subsequently with a MVA vector vaccine. And in this way, in the green line here, you can see we maximally enhance the response after the boosting vaccine and also increase the breadth of the response as shown on this right-hand plot here. However, when we then applied these same vaccines to patients with chronic hepatitis C virus, where we first suppressed the HCV viral load, we saw a very different pattern. In fact, in most patients, there was no response to these vaccines at all, whereas a minority had a low-level response, and only in a couple of patients did we see the very high-level responses that we previously observed in healthy volunteers. And therefore, it was probably no surprise that when we then applied these to patients with chronic infection where we had not first suppressed the HCV viral load, there was no effect whatsoever on HCV viremia as shown in these dashed lines in 4 patients here. In an attempt to further enhance the T-cell response with viral vectors, we've been developing class 2 invariant chain as a genetic adjuvant. So class 2 invariant chain is a type 2 transmembrane glycoprotein that sits in the peptide binding groove to stabilize MHC class 2 molecules and targets the antigen to endosomal compartments. So the rationale here was that we could enhance CD4 T-cell responses when we attach class 2 invariant chain to our T-cell immunogen of interest. In fact, we went on to show that this approach would enhance not only CD4 T-cells, but also CD8 T-cell responses. We published this in 2020. The left-hand plot shows individual healthy volunteer data with these enormous T-cell responses using these genetically adjuvanted vaccines. The group data is presented on the right-hand side, where we showed on average a 3-fold increase in the T-cell response using class 2 invariant chain as a genetic adjuvant. So the lessons that we've learned from hepatitis C virus vaccine development that we can now apply to hepatitis B is that AD-MVA vector vaccines used in heterologous prime boost are an excellent technological platform for safely inducing high magnitude T cells. That class 2 invariant chain is a potent genetic adjuvant that can further enhance T cell responses. However, crucially, even potent T cell vaccines cannot overcome T cell exhaustion that is well described in chronic hepatitis C virus infection. And T cell exhaustion is probably also the major barrier to HBV cure for those of us working in the immunotherapy field, with the well-recognized paradigm now of the upregulation of multiple checkpoint inhibitors, of which the upregulation of PD-1 is probably the best characterized in chronic hepatitis B virus infection. However, we have been inspired by woodchuck model data that emerged from the Rosenberg lab, where this group used a T cell vaccine alongside a checkpoint inhibitor, having first suppressed woodchuck viremia. And only in the group that received the triple combination were they able to show that there was a sustained suppression of woodchuck viremia associated with the loss of woodchuck CCC DNA, as you can see in this small plot down here, and with a restoration of the woodchuck-specific T cell response. So this is a proliferation assay where you see the restoration of the T cell response after entecavir is stopped. So our approach now is to make HBV viral vectors and combine this with anti-PD-1 for use in human studies. So this is a cartoon that describes the HBV viral vectors that we have now made, including a chimpanzee adenoviral vector and an MVA viral vector that encodes almost the entire HBV genome, including HBV core, polymerase, and S antigen, with class 2 invariant chain included as a genetic adjuvant. We've encoded a genotype C HBV immunogen, having first aligned all the available genotype C sequences to generate a consensus sequence, and then select a patient sequence that's closest to this consensus. And we've elected to use a genotype C immunogen because this is a huge area of unmet need, and because this is found in Asia, where we thought we would most likely be conducting our phase 1 and phase 2 HBV human clinical studies. We first performed a series of mouse experiments. I'm presenting a single slide here, which shows the HLA-A2 transgenic mouse data, which showed that we can induce a multi-specific HBV T cell response in these transgenic animals when you use a heterologous prime Bruce approach, with T cells here against HBV core, polymerase, and S antigens. Since this, we've moved on to phase 1A and phase 1B human studies. So the phase 1A study started in February 2020, which was dreadful timing just before the global pandemic hit. And I'm proud of the fact that in spite of this, we've been able to progress this work. We gave first a single Chad HBV vaccine in a 2-dose escalation study to healthy volunteers and more recently to HBV infected patients. The phase 1B study has also started. It began in February 2021. The idea here is to vaccinate 46 patients who have chronic hepatitis B in South Korea, Taiwan, and the UK. We're excluding patients that have got advanced fibrosis or cirrhosis. We're excluding patients who have got hepatitis B surface antigen levels greater than 4,000 international units. And in a subset of patients, we're also administering anti-PD-1 at 0.3 milligrams per kilogram, which has been shown to have receptor occupancy of more than 90% out to 48 days. And so far, we have randomized 30 participants. This is the study design here, 16 patients divided each in 4 groups. So the first group of 16 are going to receive 2 MVA HBV vaccines 28 days apart. The second group of 16 will receive a Chad HBV vaccine followed by an MVA vaccine 28 days later. Group 3 and group 4 will receive heterologous prime boost, either with anti-PD-1 given at the time of the boosting vaccine or with anti-PD-1 given at the time of both the priming and the booster vaccine as represented here. And we'll be presenting the immunology data from this at the American Liver meeting next week. And I apologize that I'm not able to present this today. So I would like to thank everyone who participated, especially Vaccitech, who've been fantastic partners, led by Tom Evans, and also the HBV T-cell vaccine team at Oxford, including Senthil, Tamsin, Tony, and Paula. Thank you very much, everyone, for listening to me today.